Okay, so it's the October ACDN webinar with Brad and Lynn for Canines for Wildlife.
Okay, thanks very much for having us. Really a good opportunity to share and talk to people about scent detection dog work and what we're all doing. So to start with, I'd like to introduce the team. So our people team is Jack Nesbitt. He's our dog trainer and detection dog handler. And then Brad and I are ecologists and detection dog handlers as well. Oh, hang on, I can't get it to move through the next slide. Here we are, down here.
Can everyone see the slides?
Can everyone see the slides?
Yeah, yep. We can all see them.
All right, okay, good. So this is our dog team. We've got 4 dogs working in the team at the moment. We've got 3 English Springer Spaniels and 1 Border Collie. They're all working line dogs, and all of the Spaniels have a family history of working in scent detection work. So, for example, Ash, the black and white dog, his grandfather was at Macquarie Island back in the early 2000s. Bunya's father and brother are currently at Lord Howe Island doing rodent detection, and Max's father and aunt are both scent detection dogs as well. And all of the dogs work on several odours. So there was a sort of interest expressed in the Anacondas work. So we thought what we do is just go through a couple of the projects that we've been working on over the last couple of years. So we'll touch on the Anacondas work and also some of the turtle work that we're doing. So the Anacondas work, this is working on 2 endangered Anaconda species. There's the they're both montane Antechinus, so they're high altitude. Sorry,
yeah, I'm just going to ask there's some called Gillian's iPad, I think. If we could just put that on mute. Yeah, perfect, thanks.
Thanks, no worries. So with the 2 species we've been working on is the Antechinus arctos, which is the black-tailed Antechinus, and Antechinus argentis, which is the silver-headed. They're both, for people who may not be familiar with Antechinuses, they're a short-lived insectivorous marsupial. The males live for a year, the females live for 1 to 2 years maximum, which makes them a challenging species to work with. We've been working with the Queensland University of Technology for about 3 years on these species through Dr. Andrew Baker and with his students, and also with the Queensland Government Environment Departments. And the current project is funded through the National Environment Science Program in partnership with the University of Queensland, QUT, and the Queensland Government. So the first one is the silver-headed Antechinus. So it's a small Antechinus. It's got 3 isolated populations in the rangeland, on top of the mountains, west of Gladstone and sort of Rockhampton area. They weigh around 20 to 50 grams, females around 20. They're ranked in the top 20 Australian mammals likely to go extinct. And these little fellows are ground dwelling and also arboreal. They're potentially impacted by climate change, changing fire regimes, pigs and feral cats. And the numbers are relatively low in the field. So we've been working on these species for 2 years now. The first surveys we did was last year. And the dogs are surveying for presence and absence. They're helping to ground truth habitat modelling, habitat usage and territory niches within habitat, and sort of picking up some of the behavioural attributes. For example, when they climb up a tree, that they actually spiral around the tree, and the dogs were picking up on the spiral movement of the odour going up the trunk of the tree. So we were able to indicate on individual trees that the species were using. This year we did further surveys across Blackdown tablelands, Bunburraban and Croombe, and also a couple of other small reserves. And we covered 53 sites looking for these Anacondas. We confirmed their presence at 13 of those sites. And we found 11 new locations where they hadn't been previously recorded. Some of the sites were successfully trapped post-dog indications by either Elliott trapping or camera trapping. And the data that was collected is going to be used in part to revise the habitat model that we were working on. A couple of the sort of highlights that came out of that survey for us was, one was a couple of examples of sustained territory use by the females. There's a theory that maybe the females inherit territories from their mothers. And at a couple of sites we had instances where the dog had indicated in 2018 on a particular tree or a location. And then this year we had dogs indicating either on the same tree or at the same locations within around a 10-metre radius, and also had Elliott captures at one of those sites confirming a female caught at the site. So that sort of fine, detailed information we thought was really quite interesting, and be interesting to see where that goes further down the track. One of the other highlights for us was, again, having the dogs finding locations where there's been a lot of Elliott trapping over the years and they haven't been able to capture individuals in trapping. And on one of the sites, we got 3 indications on the site by one of the dogs. They put cameras on all of those 3 indications, and at 2 of the indications, they got a single individual captured on the camera trapping post-indication. What was really interesting about that was that the cameras were out for 2 months, and when they collected the cameras, they had a single photo image of an individual 1 month after the dogs had indicated on one camera and 2 months on the other. So there's a long delay in getting a visual image of the animals post the dog's indications. Part of the problem for these species is that there's lots of other species that are jumping into the traps and running under the traps. So when you've got an animal that's very rare, the probability of getting them into these physical captures is quite low. The third point that was really useful on this particular species is that there's been a theory around for some time that they should be in rainforest as well as open forest. And despite a lot of Elliott trapping over the years, they haven't been able to confirm them in rainforest. In 2018, we had one of the dogs indicated on a rainforest site. And then this year, the dogs indicated on the same site and also on a number of sites at Bellingen in rainforest. And we also got multiple captures of Antechinus in Elliott traps at Bellingen as well, which now confirms that in fact they are both in open forest and rainforest. And that was really, is a really nice outcome ecologically, because one of the concerns is that these areas were part of the big wildfires that went through Queensland at the end of last year. So they're very fire prone in the open forest. And if they are also to be found in the rainforest, then potentially they have a fire regime and potentially some climate change refuge in the rainforest. So that everyone was pretty happy to get that confirmed. And all of the data that came out of the survey is, as I said before, going to be refining the habitat model for further work. The other one is the black-tailed Antechinus, Anacondas arctos. It's a bigger Antechinus, although it's still quite small. These guys are around 50 to 120 grams in size. They're only ground dwelling, they're not arboreal. And they're also in the top 20 Australian mammals likely to go extinct. This species is only in the high altitude rainforest on the scenic rim of New South Wales and Queensland, so very limited distribution, and also likely impacted by climate change, feral cats, and potentially fire, as we witnessed with the Benabara fires that went through the week after we left that site. We first started working on black-tailed Anacondas in 2017, and this was the first Anacondas that we tried using scent detection dogs on. In 2017, we trialled the dogs at Springbrook National Park, And we spent some time confirming the dog on they've got permanent grid trapping sites there, and we worked with the researchers trapping animals and confirming that the dog was indicating on the black-tailed Anacondas and not on the common Antechinus, Antechinus stewartii. And once that was all confirmed and we're all happy, we then started working the dog in other locations, and the dog identified 3 new locations in proximity to that trap site that they'd never trapped before, with quite a number of indications in 3 loci. The honours student put cameras on those 3 loci, and we got 100% confirmation of the black-tailed Antechinus at those sites. So then we were pretty confident that we were on the right track and that the dogs were accurately indicating on the black-tailed Antechinus and nothing else, which was really promising. Following that, we went down to the border ranges, and there've been, despite intensive elliot trapping and searching in the border ranges, there'd been no evidence of black-tailed Antechinus in over 30 years. There was a permanent grid site, and they were camera trapping and elliot trapping and not picking anything up. We took the dog down and ran the dog, and the dog indicated on 2 sites in a week. One site was adjacent to the long-term trapping site, and the other was at a historical site. We then put camera traps on those indications, and the adjacent site to the long-term trapping, we had confirmation of an Antechinus within 24 hours. A confirmation of the record on the historical site at the end of last year, so 18 months post the dog indicating. So this was a big, so these are the first confirmed records of the black-tailed Anacondas in New South Wales in over 30 years, probably due to a summer drought and lack of, they missed about 800 mil of rain in the summer. And prior to us going into the field, the PhD student spent 3 months trying to capture our Antechinus. Sorry, I'll just stay back here for a sec. So when we started the survey at the beginning of August, we didn't have any fresh animal odour to start the work on, so we were relying on the odour that we trained on, that we had in the fridge. The dogs successfully successfully identified 11 new locations for Anacondas arctos where there haven't been previous records, and that was across Springbrook and Lamington National Park. Some highlights of this particular program were that we had 41 sites that we surveyed. We got 16 positive sites all up from the scent detection dogs, and 10 new sites, not 11. And the researchers were trapping using elliot traps at the same time that we were doing the dog surveys. They only were able to capture black-tailed Anacondas on 2 sites, and they got, on one site they got 2 individuals, and that was one of the sites where the dogs had previously indicated, and they caught animals in the elliots at the 2 indications of the dog site. So one of the things that became clear with this particular survey is we were doing a lot of remote work, so we were actually camped out on the rim for 5 days, so it meant carrying a lot of gear out and staying out remote. And that's really challenging for elliot trapping because of all of the heavy equipment and the long distances you've got to carry the gear. And the dogs were able to cover a lot of sites more readily than the people who were trapping, and were more effective at picking up presence of the black-tailed Antechinus in really low numbers. So that was a good outcome. One of the challenges of working with Antechinus is the odour. Trying to get good odour to train the dogs on and for use as confirmation in the field is difficult. We've got very low numbers of individuals likely to be caught at any one time, and the survey period to be able to trap for them is only 1 to 2 months a year. So there's a very narrow window in which the researchers can support us with providing us with material for training. There are no Anacondas, other of these species, in captivity, so we're reliant on animals in the field providing us with our training odour material and our refreshing odour material. We use scat, fur clippings, anal swabs and the holding bags of the animals as to create the odour profile, and we store it in the fridge for training. Once we're in the field, we try to capture an animal or two if we can, and then get fresh material to, if you like, close the loop at the beginning of each field trip. For the black-tailed Anacondas, we didn't get that until a week into the field trip, but the dogs were still indicating well, which was really impressive given that the odour for the black-tail was actually 2 years old, because they weren't able to get any fresh odour. So we actually had odour from 2017 that the dogs actually effectively picked up animals on, which was pretty amazing. The other challenge is that these animals are very small and they don't leave any visual evidence for the handler to see. So there's no way of being able to actually visually corroborate the dogs when they're indicating. So it's a classic example of the old saying, you have to trust your dog. The other challenge with these species is that they are coexist with other of the same genus. So the Anacondas argentis and Anacondas flavipes are both in similar habitats, but there's a slight altitudinal gradient separation. But again, you've got to be really clear that they're indicating on one and not the other. And Anacondas arctos, the black-tailed Antechinus, are in the same habitat, and the brown Anacondas is really common. So again, you've got to be able to be sure that your dogs only indicate and the brown training that we have to do with the dogs. So moving on to the turtles now. We've been involved in the Bell's Turtle Project, the Turtles Forever project with the Northern Tables Local Land Services and around for a few years. This project is facing a high risk of extinction. It's a short-necked freshwater turtle that's unique to the upland streams of the Namoy, Guida and Border Rivers catchments. It's an aquatic turtle, so the female only leaves the water to lay eggs in the breeding season, and the females don't breed until they're at least 18 years old. So there's a long time delay from hatchling to adults recruiting back into the population. The biggest threat to these turtles is fox predation of the nests. Over 95% of the Bell's turtle nests are raided each season, which significantly reduces the number of hatchlings getting back into the system. The breeding season's from October to January, and full events, and so that's when the dogs are in the field. Triggered by rain trying over the top of the nest. Again, we have an odour challenge with this species because we're looking for buried eggs under hard ground. You've got a diffuse, a very light odour that's underground, hard to find. And initially, we didn't have any Bell's turtle odour to work with, so we had to work on surrogate odours to start with. And then as we've moved through, we now have a store of Bell's turtle eggs material from the previous season, and we've also been using hatched Bellingen river turtle material, a captive breeding program down at Taronga. Once we get into the field in the beginning of the season and we find active nests, then we can use those nests to close the loop in terms of using the live nesting turtle odour. This is just an example of, like I guess all of us, we use GPS tracking collars on the dog so that we can work out what the search area is and make sure that we've not got gaps, etc. On this particular map are a nest site. The flags the dogs are working over. one of the neat things that has happened over the last 2 seasons is at a number of sites we're getting really nice corroboration between the nest sites in multiple seasons, and so we've picked up a couple of sites where it looks like the females have a real preference for nesting, and this year we're going to try and actually nest protect the whole beach as opposed to individual nest sites, which hopefully will increase the number of hatchlings that make through to the next season. So just in terms of that program, in 2017-18, the dog team surveyed 22 sites and found 104 nests, of which there were 3 unrated nests that we were able to protect. And out of those nests, we managed to hatch 52 hatchlings, which went back into the river. In 18 and 19, the team covered 222 kilometres of search area and located 194 nests and found 17 live nests that were protected, plus 5 hatched nests at the end of the season that managed to get through without being predated. So that was a good increase in the numbers of nests that were found. We've got a very narrow window of opportunity to protect these nests before the foxes get them, so it's critical to get on the ground quick when the rain comes through. So the season's just about to start. It's due to start in November, and we're basically, it'll get triggered by rainfall events. So we're preparing the dogs for this project at the moment. Very quickly, just the other sort of projects that we're involved in. Ash is currently working on koala. So now that he's finished his Anacondas work for the year, he's switching over to koala at the moment. As many of our ACDN members are very effective koala people and have really good detecting dog teams working on koala. They've proven that it's a very efficient and effective way of searching for koalas. It's been clearly, they're particularly good at confirming absence, finding scats in places that demonstrate hard for people to visually identify. Really useful for presence, absence, breeding activity, disease, genetics, amongst other things. So really effective. Ash is working on koala scats at the moment, and he's back in training on that as we speak. So that's basically a quick overview of the sort of work that Canines Wildlife are involved with. We're really pleased with the results that we've got with our dogs this year. They've proven to be really resilient in the field under really tough conditions and have been versatile in working effectively on several odours. They've provided real value to the research team work that we've been collaborating with and to the conservation projects that we've been involved with. And it really confirms for us that dogs have real value to contribute to research and survey across a broad range of activities in the environmental sector, and we're currently looking for new opportunities to expand into and explore with our dogs. So thanks very much for listening to us, and over to you guys for questions if you have any.
Thanks for that, guys. That was very interesting. I'll just open it up then. If anyone does have any questions, just go ahead and unmute yourself and ask away. I know that I have one actually. I'll get going. I was just wondering, with the Bell's turtle and that kind of the fox predation there, what are they doing about the foxes out there, or do you guys assist in that way with dogs as well?
At this stage, there's 32 properties that make up the survey sites, and each of those property owners are able to sign land services for funding for fox control, and so some shooting and to a small extent baiting. A lot of the properties are sheep farmers, and so they have their own challenges in terms of using 1080 baits when they've got working dogs on the property. And so most of our focus around trying to control foxes is really about getting protective measures right on top of where the nests are. And then now with the last season's work, it's clear there's priority sites where turtles are returning to nest, And so we're looking at protective measures along those beaches to try and protect more nests with less infrastructure. But it's a challenge because a single fox, we've watched them at different times moving along the banks and with the vixens with their young cubs. And you'll see them actually training as they work and they scavenge along the banks. And they're actually getting the eggs at times. The eggs have been laid early in the afternoon and they're eaten that night by foxes. So the window for us to get out there with the dog quite narrow. And in addition to that, foxes at times are actually catching the adult females as they lay and then killing them and then extracting the eggs from their abdomen. And so that's not only tragic for that turtle, but when it takes 18 years for them to reach maturity to lay eggs, and they can live for a long period of time after that, the breeding capacity that's lost every time one of those individuals disappears is devastating. And so there's a lot of thought and a lot of effort that goes into trying to increase the fox control, but it's really challenging in these pastoral landscapes to be able to do it effectively.
If I might, a word with regard to fox control. Where the eastern part of Kansas in the 1960s, right when I grew up, but there were 4 species of fox native to that area, and they were virtually exterminated because of the fur trade and the depression. And how that worked was there was an increasing economic incentive as the animals got rarer, the law of supply and demand. And when I was a kid in the 60s, if I could have caught one, an individual red fox would have been worth $50 to $100. And I desperately wanted to catch 2, because then I would have enough money to buy a horse. And but I was too late. Try as hard as I could. I could not catch one. But they say the trick is getting an escalating bounty in the right places to extermination. And the way that I couldn't figure out a way to do that, round up here quickly, but thanks to CRISPR, we could now do that. We could, I identify the genes of the foxes that only live where the Bells River tortoises are, and offer higher and higher incentives for the farmers to have successful fox control programs. Then they would try harder. They'd go out and try. It was just a small incentive at first. You go out and have a go, and if they were successful, we could offer more. The LLS could offer more and more money as time progressed for the population around turtle. The Victorians tried. Rewards don't work because people cheat inside the area where the rewards are supposed to come from and bring things in. I know I used to cheat with coyotes. I tried to get rid of them. And you guys might be in a position to suggest that. We think you might succeed more with these turtles if you gave the landowners some genetic work on the foxes and gave a ever increasing reward for landowners to kill foxes, to kill Bell's turtles.
Yeah, it's an interesting idea, Jim. Thanks for that thought as an escalating bounty, and it's certainly not something that I've heard of being in Australia, and with some genetic research support to be able to make sure that you're focusing on the right individuals. It sounds like an interesting idea to run with. There's also some thinking around foxes, especially when they first came into Australia and we would have had much healthier turtle populations, that they may, as you come into the breeding season for turtles, because it's not only Bell's turtles, it's the Emiduras, the long-necked turtles, other species, they're all nesting around the same time. And that there may be a behavioural response by foxes where they come in from further afield two locations where there's a plentiful supply of food for the season. So there may be other dynamics at work as well. But I think we need to broaden our toolbox from not only trapping and 1080, but escalating bounties and other techniques, because there's no one fix to address this issue, and I think we need to be trying everything that we can. So that's a great suggestion. Yeah. Thanks for that.
Well responded to.
I was just going to add to that, the other thing that's really cool is doing outreach to tell the story about your turtles, which I'm sure you're already doing. That has been useful for leveraging more funding and more community support in our experience as well.
Yes, yeah, thanks. And there is a bit of work that's being done in that space as well. There's also a PhD and some masters research as well with schools, and it's quite a large program and very. And the outreach doing is really comprehensive and warrants another presentation in its own right. Thanks.
One interesting thing I learned from your presentation was and you guys learned it because you had to, was the length of time scent will last in the refrigerator. I've been refreshing my koala scent, which I suspect is much stronger than that. I only keep it for a month or so. Can be a big hassle, not so much for me. I've got a ready source, but other people that I supply with it. And so that was very interesting to know that was good to get that out there.
Yeah, and we were very surprised that the 2-year-old odor was effective. We were hopeful, but not overly confident that it was going to be able to get us there. Yeah. So it was, a great relief that the dogs actually were able to make that connection across. So it really is interesting in terms of that whole aging and holding of material. It's an interesting area to think about, because it's a challenge we all have with all the odours that we work with, unless we've got a ready supply of something in captivity.
Yes.
I'd just like to add to that. It's Emma here. I keep some of my samples for years and years and years, and then I go out and test people with them, and the dogs do just as well.
Confirmation.
Yeah, that's good to know.
And was it with those samples that you guys had, did you manage to get it from many different individuals, or was that a challenge as well when it came to the Antechinus?
That's a real challenge, because there aren't many individuals to sample from. So I think we're more of the silver-headed than we have of the black-tailed, but you're talking, I can't remember, Jack, exactly how. I think they caught 15 all up of the silver-headed across all of the sites, something in that order. So you're looking at less than 20 individuals across 3 populations. And for the black-tailed, as I said, this year they caught 3 individuals. So that's the material we have going into next year from this year. But the dogs seem to be able to make that bridge okay.
Yeah, I think more of a I don't know, an observation than anything else. The data that you guys and the refinement of the trapping programs that you guys were able to inform dogs is mind-blowing. I mean, it means that management strategies in areas where they didn't think they had it, now they can actually know they have it and treat the area differently. It's just brilliant. Well done. Congratulations on your hard work, and it's why I'm in it. And yeah, I think you guys are phenomenal.
No, thanks very much. Yeah, look, it's been really exciting, and again, great collaboration with the universities, and the Queensland Parks and Environment Science guys have all been really, really on board, and have really done the hard yards to help us to get the dogs out there, particularly on the remote sector that we had to work on. That was a bit of an interesting challenge. But Yeah, it's really interesting to keep pushing forward and seeing what the dogs can offer in this space. And it's great because we've now got a PhD student, Stephanie Batista, who's working with us on detection strategies for endangered small mammals, and she's focusing a fair bit on the detection dog side of things. So it's really it's exciting to be working with young students and cutting edge science. Really, yeah, it's very fulfilling, if not challenging at times.
Yeah, well, I worked in programs down in Victoria where we had to look at, and one of the greatest risks we had was a lack of data, because you make decisions and biases based on the data you have, and when you have great amounts of data missing, then this is where dogs can provide a different data set and more information. So yeah, it's awesome.
Yeah, I think one of the interesting things for me was with some of the silver-headed stuff, we were able to cover, in one instance, a significant portion of the whole park. So we spent a fair amount of time outside of modelled habitat, and then worked in high and moderate and low habitat as well as non-habitat. And so working the dogs provided an efficient means to be able to cover a significant amount of territory in a relatively short period of time, which the people doing the trapping just can't do. So they really provided another information tool to really refine that habitat modelling that they wouldn't have been able to do without the dog work. Yeah.
I'm reminded of a comment Hugh Possingham made once when I commented on the model of habitat that he had made for the Hastings River mouse. They said, but Hugh, you don't have any data to go into that model, and it's not going to be much use to me in making national parks. He said, no, Jim, you mistake the purpose of models. Models are not for designing national parks, they're for frightening politicians. And so his models always show everything going to extinction quickly, rather than where we should put the habitat. So I'm sure that he will come around. It was that he didn't have any good data available, so he was trying to do the best thing by way of the environment. But this offers a whole new level, and you guys with the enthusiastic young students. I'm interested in the remote area. How remote is remote? Did you have to hike into the spots?
Yes, we did. We had to walk in about I think it was about 8.5 kilometres to the base camp,
right?
And then we worked, which is not that far, but we had to take all of the food and water for the dogs, plus all of the Elliott traps, and we spent 5 days out on the rim and surveyed remotely from there. And the dogs were doing up to 20 kilometres a day circuits from there, plus doing, in addition to doing the actual searches.
Right.
So yeah, it was an interesting, we hadn't worked quite like that before. So that was an interesting challenge. We've learned a few things out of that. The dogs actually handled it surprisingly well.
Yeah. Well, having a dog walk along beside you is a heck of a lot easier than carrying boxes.
Yeah, I think the people who are doing the Elliott trapping would agree with that, for sure.
Roman, are you able to mute your video? We're just getting a little bit of interference. If you can hear us. Yeah,
Thank you.
Thank you. Yeah, that sounded like a challenge, but quite an exciting one. And I bet you the dogs handled it easier than the humans, huh? 20 Ks a day, and
Yep, I'd say that's true. That's where having Jack on the team makes a big difference too. Having young legs makes a big difference.
Young legs.
Yeah. Super.
I had a
Yep.
around training. So we're doing some turtle nester training for some of our dogs as well. With the same sort of problems, how do you get enough samples, and how do you make sure that the volume of samples is enough? Have you thought about using any other type of sample? So we're looking at, just out of necessity, using nest plugs. So the soil that is capping the hole the eggs are laid into. Is that something you've branched out into when training for your dogs?
Yes, so we did last season. So we found, the dogs found a nest that had plug material, which was in like a clay loam site, whereas a number of our sites are actually really fine river sands or coarse shingle, and so you don't get any plugging there. But in this particular area, there was a clear plug, so we collected that and then added that to the odour profile, and that was really in that particular substrate. And we've kept some of that material, so we're using that as the pre-season training. And we're looking this year to also collecting as part of the early trapping that will occur to determine when the females are becoming gravid. And so when we might expect nesting to occur, and then focus the survey effort with the dogs of collecting ballast water from the turtles when they're trapped, with the idea of that because part of that would certainly be deposited when eggs are deposited, and then adding that to the profile as well. But it's the challenge, Nick, getting the odour. Like the dogs work really well with the tools that we give them, but it's trying to get the right odour at the right time as well. Yeah.
I have a question around that too. Are you freezing or are you refrigerating your samples?
A combination of both. When we have eggs from the previous season, especially infertile eggs, that because as part of this program, some eggs are collected and incubated, and those turtles are then hatched and then released back into the wild. The infertile eggs, as part of that part of the program, some of those are provided to us, and you just can't keep those for 12 months and expect that they're going to be useful. So we freeze those and then defrost them, because otherwise we'd be training them on a decaying odour profile, not a fresh turtle odour. So a combination of both when we can. We're also using the Bellingen river turtle because it's the same genus. They're both mycelids. And very kindly, the people who are running the program at Taronga are providing us with the vermiculite that their eggs are hatched out in, the soil that they actually lay their eggs into, again in fertile eggs and hatch. And although it's not exactly the odour that the dogs are looking for, in establishing a broad odour palette, which we then fine tune as quickly as we can when the season starts by finding live nests and then focusing specifically on that particular odour.
Cool.
Excellent.
Turtles, I reckon, are one of the hardest. It's just constantly giving us headaches, really, to make it work. But we keep getting right each year, so it is working. But it's the turtles.
A huge slog. But like you said, so valuable when you do get those wins. And 17 is incredible. 17 new nests is awesome.
It's amazing.
Yeah. Especially when you multiply that by 20 odd eggs, and then you start to get good numbers. Yeah.
Awesome.
Something for the community outreach and liaison as a means of reacting to the new information that you guys have got. I'm the scientific advisor to a local group, the Australian Conservation Hunters Association. And they're people who like to hunt mostly deer, but they are knowledgeable bush people, and they're very active. And hopefully some of them will be watching this webinar later. I know they put it out to the whole club. And I guess what I'm point of making a comment here is they're pretty useful. They go out and spend their money doing this stuff anyway, and they are willing to. They're interested in wildlife and conservation generally, but they're tarred by the brush of being shooters and hunters. And I bet these guys to at least change their name from shooters to hunters. They're not target shooters and that sort of stuff. They are bushies who want to have a love, and they are interested in conservation. So, yes, our public liaison lady, I guess I'm talking to. I apologize for a terrible name for applying for names, but a bit of reaching out to them and just seeing if there's a group like that in. I know there are conservation hunting groups in the New England area, and so I don't know. That's a suggestion for future action.
It's a good point to make, Jim, and it makes me think that there was one property we went to last season. We're surveying, talking to the property owner, and the conversations I always have is around like, what sort of control have you been doing? What success have you had?
Yeah.
And this particular property owner, it's quite a large property,
yeah.
but he had a number of shooters who came on during the year, and they'd shot 300 foxes on his property.
Yes.
In the year. And you think that would make a significant difference, yet foxes had raided every nest that the turtles had attempted to lay.
Yeah.
And so it's that challenge of getting the right foxes and the highest in that nesting area. And that's where certainly focus and pressure by people around those locations. I don't know where his 300 were taken from. They may well have been out on the open flat, necessarily in the drainage systems. But it just strikes me that foxes out of the system, but still not as many as you need to.
That's right. That's right.
Yeah, they're very cunning, and they can do a very, very short period of time. We know that from down on project down in Warrnambool in one night.
Yeah. Possibly if you could concentrate our control temporally as well as spatially, you guys have found the control needs to be done, and you know when it needs to be done. And maybe you could liaise with the LLS, and I'm clear down here on the southern tablelands. These guys I know drove to go fishing, drove to Queensland, and I don't know. The suggestion, see if it happens.
Yeah.
And Ramon and her lab do this CRISPR stuff really well. As far as getting the genetic pool of the foxes we want to get rid of, might get her involved.
Yeah, that's a great suggestion. Yeah.
I'm great at coming up with stuff for other people to do.
All right. Just of the time, guys, it's 7 o'clock. We give everyone an hour for this, so unless anyone has questions, I was just going to say thank you very much to Lynn and Brad for a very interesting and inspiring presentation, and to also quickly let everyone know that in December we're going to have Latoya and Naomi from Zoos Victoria, who are going to be talking about the detection dog program that they have in Zoos Victoria, and also I think a bit about their recent trip to New Zealand as well. So we've got so we'll stop the recording for now, and then we can, if anyone has any general business, we can